Good morning, everyone. I am Thachamonthon Petborom, fourth-year dental student from Chulalongkorn University. Today, I'm here to talk about an interesting study in field of dentistry called Lactobacillus plantarum Lipoteichoic acid inhibits oral multispecies biofilm. This study is about inhibition effect of lipoteichoic acid extracted from Lactobacillus plantarum on multispecies oral pathogenic biofilm, which consists of Actinomyces nasalizii, Enterococcus faecalis, Lactobacillus cerebrius, and Streptococcus mutans. First of all, let's start with introduction. Biofilm is a community of microorganisms which is generally found on moist surface. Living as biofilm improve ability to survive, express outer genes, and resist antimicrobial agents and host defense mechanism. Biofilm on two surfaces may cause inflammation and diseases. For example, biofilm on root canal wall is the cause of apical periodontitis. Therefore, many researches were performed to determine the way to reduce biofilm as much as possible. One of them is using Lactobacillus plantarum. Lactobacillus plantarum is gram-positive bacilli, facultative and aerobe bacteria, which is known as a probiotics. Its cell wall contains lipoteichoic acid or LTA, which attach cell wall to cell membrane. Previously, there are researches about L-plantarum lipoteichoic acid or LP-LTA inhibition of one bacterial species, but there is little known about inhibition of multi-species biofilm. For that reason, this paper was emphasized on multi-species biofilm inhibition and interference by using LP-LTA. The aim of the study was to determine if LP-LTA can inhibit oral multispecies biofilm formation and perform multispecies biofilm. In addition, this study was determine if LP-LTA combined with conventional intracanal medicaments, including clozidine digluconate or CHX and calcium hydroxide or CH, can disrupt perform multispecies biofilm. The methodology of this paper was divided into many parts. Start with LTA purification from L. plantarum. LTA were extracted from L. plantarum cell wall and purified by dialysis. Then it was collected, freeze-dried, and measured for dry weight. Next is multi-species biofilm formation. 4 bacteria species were grown in different agars and put into PYGA broad media. Then it were mixed together and cultured. The experiment was divided into 2 main sections. The first section is about LP-LTA inhibiting biofilm formation, and the second one is about LP-LTA disrupting perform multi-species biofilm. The first section was subdivided in 3 parts. First, beginning with anaerobically culturing of mixed bacteria on glass bottom dishes with or without LP-LTA in different concentrations. Next, staining slides with cyanide and propylene iodide, and then observed through confocal laser microscopy. The next part, bacteria were grown on 96 well plates in the same conditions, but it was stained with crystal violet and measured by microplate reader. The last part, bacteria were cultured on the thin slices under the same conditions. These thick slices were examined by scanning electron microscope. The second section starts with growing mixed bacteria for creating biofilm on glass bottom dishes, followed by adding varied LP-LTA concentration alone or adding with CHX or CH. Then the slides were incubated following by staining with cyanide and propylene iodide. It was observed through confocal laser scanning microscopy. After collecting all experimental data, mean values and standard deviation were calculated from 5 samples per group. Difference between groups were analyzed for statistical significance at p-value equals to 0.05 using the non-parametric Crutchlow-Wallace and Dunn test. Next is results. This part was split into 2 sections as the same in methodology. Start with the first section, multi-species biofilm growing in glass bottom dishes, 96 well plates, and human dentin slices gave the same result. Newly formed biofilm with LP-LTA added were significantly less than the non-added one in a dose-dependent relationship. For the second section, the results shown that the preformed biofilm on glass bottom dishes with LP-LTA and CH or CHX were significantly less than the one with LP-LTA alone and the control setting respectively. Now we are moving to the discussion. LP-LTA have ability to reduce multi-species biofilm on both new biofilms formation and existing biofilms in a dose dependence, and it also enhances the ability of chlorhexidine digluconate and calcium hydroxide. LP-LTA has been suggested as it plays a role in anti-inflammation, so researchers believe that LP-LTA not only enhances those two intracranial medicaments, but it may also reduce inflammatory response. In our opinion, this research still has no evidence of experiments in animals or humans. In vivo studies should be done to ensure the results and safety in practical use. In addition, the experiment was conducted only in dentin slices. In the future, studies on enamel slices should be performed because biofilms in normal oral cavity attaches on pellicle coating on enamel surfaces. Finally, we come to the conclusion. This study showed that LP-LTA can inhibit formation of multi-species biofilm and also reduce the preformed biofilm. Furthermore, LP-LTA can improve the abilities of calcium hydroxide and chlorhexidine digluconate. And here is our reference. Thank you for your attention. Goodbye.